Let's talk about antigens.
First, let's have the definition of terms. Immunity is defined as resistance to disease, specifically infectious disease. In simple terms, it is the ability of our body to defend itself against disease-causing organisms. The collection of cells, tissues, and molecules that mediate resistance to infections is called the immune system. It is the complex network of cells that mediate immunity, and the coordinated reaction of these cells and molecules to infectious microbes is the immune response. The immune response is how your body recognizes and defends itself against disease-causing organisms. So immunity is achieved through immune response by the immune system. Immunogen is defined as any agent capable of inducing an immune response. Antigen is any agent that binds to components of the immune response such as lymphocytes and antibodies. Take note that all immunogens are antigens, but not all antigens are immunogens, because not all antigens produce immune response. An example of this is hapten. Hapten is a low molecular weight molecule that alone is too small to stimulate an immune response, but can combine with another molecule to induce a response. Epitope is the portion of a molecule. Example is antigen that binds to an antibody or T-cell receptor. It is the part of the antigen molecule to which an antibody attaches itself, also known as antigenic determinant. Classification of antigen: complete antigens or immunogens and incomplete antigens or haptens. Complete antigens or immunogens, when these antigens enters the body, evokes the immune response without any assistant or carrier molecule. They possess both qualities, immunogenicity and antigenicity. Immunogenicity is the ability to produce antibodies, while antigenicity is the ability to react with specific antibody or activated T-cell. Now, let's talk about incomplete antigens or haptens. These are foreign substance which require carrier molecule to act as a complete antigen. Haptens are low molecular weight compounds which are having antigenic property but lacks immunogenic property. The immunogenic property or production of antibody is governed by a carrying molecule. The carrier molecule is a non-antigenic component and helps in producing the immune response. Adjuvants is a chemical which, when administered with the antigens, enhance or provokes the immunity. It is a chemical suspension or liquid suspension in which antigen or foreign proteins are dissolved. Since most of the antigens are proteinous in nature, they exhibit a maximum antigenicity if injected simultaneously with the suitable adjuvant. Commonly used adjuvant are Fred's complete and Fred's incomplete. Adjuvant can react in several ways. 1. Alter the distribution and persistence of antigen within positive host. 2. Stimulate lymphocytes production nonspecifically. 3. Activate macrophages. 4. Alter traffic of circulating lymphocytes. Types of antigens: exogenous antigens and endogenous antigens. Exogenous antigens enters the body or system and start circulating in the body fluids and trapped by the antigen processing cells APCs such as macrophages and dendritic cells. The uptake of these exogenous antigens by APCs are mainly mediated by the phagocytosis. Endogenous antigens are body's own cells or subfragments or the antigenic products that are produced. These are further classified into autoantigens and alloantigens. The endogenous antigens are processed by the macrophage, which are later accepted by cytotoxic T cells. Autoantigens are synthesized by the body. Examples are nucleoproteins and nucleic acid, while alloantigens are same set of molecules with genetic variation. Examples are blood group antigens and histocompatibility leukocyte antigens. Now, let's proceed to the chemical nature of immunogens or antigens. When we say chemical nature, we are talking about the chemical composition of immunogens. First, we have protein. The vast majority of immunogens are proteins. These may be pure proteins, or they may be glycoproteins or lipoproteins. In general, proteins are usually very good immunogens. Then we have polysaccharides. Pure polysaccharides and lipopolysaccharides are good immunogens, capable of eliciting immune response. Next is nucleic acid. It is poorly immunogenic. However, they may become immunogenic when single-stranded or when complex with proteins. Lastly, we have lipids. In general, lipids are non-immunogenic, although they may be haptens. Property of antigens or factors influencing immunogenicity. 1. Molecular mass. Smaller molecules does not provoke immune system. The antigens possess an optimum molecular mass of large molecule, which then binds with the receptors and provoke the immune response. The molecular weight should be at least 10 kilodalton. 2. Foreignness. The immune system normally discriminates between self and non-self components such that only foreign molecules are immunogenic. 3. Chemical composition. In general, the more complex the substance is chemically, the more immunogenic it will be. 4. Physical form. In general, particulate antigen are more immunogenic than soluble ones, and denatured antigen are more immunogenic than the native form. 5. Rigidity. Loss, alteration, and denaturation leads to loss of reactivity. 6. Spatial accessibility of determinant groups. Epitope must be exposed and accessible to the receptor to react. More exposed, more immunogenic. 6. Insolubility. Refers to the physical state of the antigen. It must be insoluble and particulate to be immunogenic. More particles, more immunogenic. Classification according to relationship to the host. 1. Autologous. 2. Syngeneic. 3. Allogenic or homologous. 4. Xenogeneic or heterologous. 1. Autologous. Antigen found within the same individual. 2. Syngeneic. Antigen found in individuals of an inbred strain identical twins. 3. Allogenic or homologous. Antigen found within the same species but antigen is supplied by different individuals. 4. Xenogeneic or heterologous. Antigen that occur in different species. Classification according to its presence in the host. 1. Sequestered antigen. 2. Blood group antigen. 3. Tissue type antigen. 4. Tissue-specific antigen. 1. Sequestered antigen. Autologous substance that do not come in contact with antibody-producing cells, since they are inaccessible. 2. Blood group antigen. Found in the surface of RBC, and in some instances in secretions. 3. Tissue-type antigen. Genetically endowed and naturally present in the tissue. Example, HeLa antigen. 4. Tissue-specific antigen. Antigen that are unique and common to specific organs. Examples are thyroglobulin from the thyroid gland and PSA from the prostate gland. Classification according to the ability to stimulate immune response. 1. Thymus-dependent antigen. 2. Thymus-independent antigen. 1. Thymus-dependent antigen. Antigen is processed by the macrophage and presented to the T helper cells. It provokes immunologic memory. 2. Thymus-independent antigen. Antigen that can activate B cells directly without the intervention of T helper. Only IgM antibodies are produced. Classification according to other serologic behavior. 1. Agglutinogen. 2. Precipitinogen. 1. Agglutinogen. Particulate and becomes aggregated or clumped. Example, IDC antigen in blood typing. Hemagglutinogen can cause the clumping of RBC. 2. precipitinogen, soluble and are precipitated or settled out. Example, CAR-P. Factors contributing to immune response of the host. 1. Dosage of the antigen. 2. Frequency of encounter. 3. Route of introduction into host. 4. Age and gender of the host. 5. Genetic endowment. 6. Underlying disease. 7. Locations. 8. Age.